Hi, everybody. Can you hear me okay? Yeah? All right, great. Thanks very much. Please have a seat, settle down. My name is Samuel. I work for Digital Selection. We're based in New York, Barcelona, and Nantes, and we do exec search for startups in digital, health, and impact industries. And I'm delighted to be a host for just an hour, and at this special moment of startups pitch session. So what we're going to do today is we'll introduce you to 3 startups innovating in oncology. And the aim is just to present new ideas, solutions, spark questions and conversations that will hopefully improve people's lives worldwide. So each startup will have about 5 minutes to present the solution to world-leading experts, you, and then they'll have 5 minutes to answer questions again from you. So please bear in mind that each of them are in different locations. One person is going to be here physically. And a way to thank them is to ask them very interesting questions. So please welcome the first startup. And we have Sophie Borzet, who's who's the founder and CEO of HICAT. Okay, so we've got HICAT Fluidics. Sophia, are you ready?
Yes.
Fantastic.
I'm ready.
The stage is yours. Good luck.
Okay. Hi, everyone. So my name is Sophie Borzet, and I am the CEO of HICAT. HICAT is developing a biomedical instrument to sort biological nano-objects for breakthrough cancer diagnosis. Today, if you have, for instance, pancreatic cancer, your life expectancy is very short, and you receive a treatment with a very high probability being ineffective in your personal case. And this is very frustrating because, in fact, there are opportunities for improvement thanks to a new kind of liquid biopsy based on exosomes. Well, now we should say small EVs. So what are exosomes? Exosomes are nanoparticles released by cells and involved in cell communication. They are present in very large numbers in all our bodily fluids. And according to several American researchers, they could well be the greatest advance in medicine since antibiotics. So that's great. And so what's the problem? In fact, exosomes carry crucial physiological information. And exosomes excreted by tumors have great potential as non-invasive cancer biomarkers. But today, there is absolutely no instrument to sort exosomes, to isolate them by specific criteria, and namely by which cell they originate from. So, of course, they carry crucial information, but researchers use them in laboratories. But in medicine, in the medical field, nobody uses exosome yet, or almost nobody, let's say. So this information is completely untapped, unused. So that's the problem I decided to tackle in 2021. And today, we have a prototype that can detect, count, and sort individual nano-objects by functional criteria, namely by fluorescent biomarkers. This is a microfluidic and optical innovation. We have already 4 patents. And what is very different in our instrument is that we have a very, very high specificity due to the very small volumes we are dealing with, a few femtoliters, which is much smaller than any droplet PCR or any cell sorter. And we also have a high sensitivity because we are going very fast. This is very important because if you want to do a statistically significant analysis on a sorted subpopulation, you need to have enough matter to do so. So we make 50,000 sorting decisions per second. Who is behind all that? Well, we are three co-founders. I am a physicist. Nobody is perfect. I have a PhD in quantum physics, and I also have 10 years experience in the industry of optical instruments. My co-founder, Mathias Giraud, is a former researcher in microfluidics, and Philippe Grindorge is in charge of the optical and electronic parts of the instrument. Our facilities are located near Bordeaux in Pessac. And here you can see part of the team 10 days ago at Institut Pasteur, where we could measure many different samples.
1 minute to go.
Sorry?
1 minute to go.
Oh,
Il reste 1 minute.
Okay, so our roadmap is very clear. In 2023, we were a grand prize winner of the iLab competition, and we could raise money. And now we are trying to find new funds to industrialize our prototype and also to find collaborations in order to demonstrate real use cases and to get data, preclinical data, and afterwards clinical data for our instruments. So what do we need? We need, as I said, collaborations. We need, of course, investors, and especially today we would like to find a key opinion leader in oncology, somebody who would think that exosomes are the future of medicine and that together we could unlock all their potential. Thank you very much.
Fantastic, thanks very much.
I'm sorry, I don't hear you very well.
Thank you very much, Sophie, for this. And we'll start now the questions and answers. And okay. Florian's going to...
Do I stop sharing my screen or... No, maybe not.
You can go back to the first slide or just leave it as it is now.
Okay.
Are there any questions?
Hello, I'm Philippe Armey. I'm from Ghent, Belgium. We have some experience with bladder cancer diagnosis. Do you also have these samples in your machine, so on urine, or it's only on serum samples that you're taking?
Well, in fact, today we have a prototype, and we focused on the essential part of the prototype. So the samples have to be prepared. So they have to be prepared with chromatography machines or centrifugation. So so far we don't care on which kind of clinical fluid or bodily fluids the sample is coming. We are at the very beginning, in fact. I don't know if I'm answering the question.
That was a yes. Thank you, Sophie. More questions? And if you want to ask the question in French, I will try to translate as much as you don't get too technical.
I will try in English. Loïc Vaugier, I'm a radiation oncologist in Nantes. Two simple questions. One about the technology which is inside your prototype. It's, I suppose, a bit complicated in terms of physics. It's like based on lasers, on something you broke from your quantum physics experiments?
No, not at all. There is no quantum physics at all. It's a kind of flow cytometer, but with microfluidics. So in fact, we use droplets, water droplets in oil. In fact, it's not water, it's the sample, and we use oil, exactly like cell sorters, microfluidic cell sorters. So we encapsulate, in fact, the sample in droplets. It's just statistical. It's just like a cell sorter. It's the same behavior. So you have many droplets with nothing inside, and the volume of the droplet is just calculated so that you have only one object in each droplet. And then we used an electrical field to sort the droplets at very high speed, so that you can have enough matter to do, for instance, proteomic analysis or whatever you want. So we think that in the future, we'll be able to demonstrate a better stratification of patients or even very early diagnosis. We do believe that this instrument could bring something really new. So, but we still have a lot to do.
Okay, and why do you focus on pancreatic cancer and not other cancers? Like, I mean, pancreatic cancer is a difficult cancer to treat, it's true, but there are a lot of other cancers like breast cancers or prostate cancer, which are very frequent. Is this, why did you focus
yes,
first
but
on?
In fact, it's just that we're working with people in the University Hospital in Bordeaux, and we are working on pancreatic cancer, and on the small EVs excreted by the tumor, there is a specific biomarker. This has already been demonstrated. So for us, it's going to be the first trial because we already have a specific biomarker. So, but we are not doctors, as you understood, and we are not even biologists. So in fact, everything has to be demonstrated, I think. We have to demonstrate that even if you make subpopulation of exosome, first you have to find specific biomarkers, and then inside the exosomes, you have to describe or to explain what is going to better stratify the patient or help you to identify, I don't know what. So there is a lot of research to do from our side for the machine and from the clinical side. This is a big challenge because, in fact, this instrument is so new, in fact, that nobody knew that we could sort nanovesicles. So everything has to be done.
That's very interesting. Thank you.
So it's a great challenge, but I think it's a great opportunity too.
Thanks very much, Sophie. Thank you, Sophie. So next on stage, we have Frédéric Nahon, who's the founder and CEO of Magix Genomics. So over to you, Fred. Thank you very much. Please welcome Frédéric.
Merci beaucoup. Thank you very much. So, just wait for my slide. So first, I want to thank you very much to give me the opportunity to introduce you Magix Genomics. I don't remember exactly how many times I was able to hear ICO. So I was trying to find the best definition for ICO today. And I think it's clearly innovation conquers the West. So I'm happy to be part of this challenge. C'est bloqué. So we did a proof of concept on soft tissue sarcoma, and we realized that we could address therapeutically challenging cancer. Challenging cancer in terms of aggressiveness, in terms of complex genetics, in terms of prognostic. And in particular in this cancer, there is no clear guidelines for chemotherapy. We also identify other cancer where we observe exactly the same as in soft tissue sarcoma. It's more or less lung cancer, breast cancer, the breast negative triple cancer, stomach cancer, ovarian cancer. We also have a lot of proof of concept beginning in some urological cancer. And urological is very, very nice to have because there are a lot if you consider prostate and bladder. So finally, what we do with Magic Genomics, we select patients responding to or at risk for chemotherapy just by analyzing the genomic instability inside the tumor. From the solid tumor, after biopsy and after the sequencing of the tumor, we are able to compute all the DNA breaks in terms of replication and in terms of transcription. And with this, we are able to stratify the population in two categories: good responder to treatment and bad responder to treatment. We have these two innovating biomarkers, and it's under intellectual property of the startup. This has been developed in Toulouse, in INSERM Toulouse, with the team of Frédéric Chibon. What we demonstrate that with a low genetic instability concentration, the chemotherapy was deleterious. In moderate, it was beneficial, and in high concentration, there is no benefits. And finally, we compare to other solutions used in the Oncopole of Toulouse, and we only have two other solutions to compete with. One is based on histological grading system, which is not stratifying a lot patient. Another one is CSARC. It is not good also for stratification of patient. And finally, we demonstrate inside our own laboratory in Toulouse that we were doing much better. Regarding our technology, there are two publications today. One is based on leiomyosarcoma, a sort of soft tissue sarcoma. And we expect for the second one on soft tissue sarcoma with a cohort of patients of 250 patients. We have today signed a partnership with Gustave Roussy too, based on 4 retrospective studies and 1,200 patients. Of course, I'm not alone in this big challenge. We are right now 6 people. You have Kathleen here in the room, our project manager. Myself, I've got a background in launching new innovation. I was involved a long time ago with Viagra, with Pfizer. And then I've been involved launching the Da Vinci system in France and Belgium. I was one of the first salary of this company. And I am happy to have this new project with all the team with very complementary quality.
1 minute to go.
We also have some prize recognition. We've been helped first with the Fonds Emergence from BPI, the French bank, public bank. We were the winner of the last edition of iLab. We've been winning 2 other prizes in Europe. And we've been recognized very recently, that was last week, in Castres for the e-Santé, e-Health challenge. Finally, innovation conquers the West. What does this mean? With our solution today, we could expect and engage together, as I mentioned earlier, on a low GI patient, what to do with those patients where the chemotherapy is deleterious. It's also to identify new alternatives for less invasive support. Let's try to work on circulating DNA, for example. It's also to try to better stratify patients with the CRO partnership of your establishment. And it's, of course, try to define a medico-economic model to try to impact and reduce chemotherapy when it is not efficient for patient. Thank you very much. And let's go together, stop the one-size-fits-all, which is today in France, more or less, and in Europe, writing the personalized medicine history together. Thank you
Thanks very much, Frédéric. Okay, let's switch to questions and answers. So who wants to start with the first question? all jump in. First question?
Thank you very much. Good question, I'm sure.
I tried to have a question. When do you start? You talked about uro-oncology. I'm a neurologist, so I know very well the problems of the genetic instability or all these castrate-resistant tumors. Do you start from biopsies? Do you start from serum samples? What is your starting point to do this analysis?
So I heard you come from Gand, is that
Yes.
correct? Are you working with Alex Motry?
No, he works in Aalst.
Okay,
so
sorry.
far away.
My
No
mistake.
problem.
My mistake. So that was not a good question.
No,
So
it's a
we
first
work
question.
on biopsy. We are involved with, of course, the French Association of Urology. We try our solution with De La Taille in Henri Mondor. And we also establish a partnership with AK Vienna Center in Austria on bladder cancer.
So you're starting from tissue samples,
Yeah,
samplings?
yeah.
Okay. Thank you.
Are you more oriented on prostate or bladder?
More prostate.
And what do you need for your technology? It's only tissues, or also it's liquid biopsies possible?
No, liquid biopsies could be the next step. Today we are working on solid tumor, and if you have in your data set RNA-seq, this is perfect for us.
And you're speaking about chemotherapy, but we have plenty other drugs in medical oncology than chemo. Did you experiment with other treatments and your?
So this is the next step, of course, is to try other chemotherapy and, of course, immunotherapy. We think we have many opportunities to develop. It's a bit strange to say we have a solution that will be useful for every cancer in every therapy. So let's go step by step.
There's a question at the back over there.
Thank you for your very clear and short presentation. I have a question regarding the aim of your medico-economic or health economic assessment evaluation. Could you tell us a bit more about that? What do you plan to do? What is the aim at the end? Thank you.
So this is a very good question. If you consider today our solution could reduce the potential patient for chemotherapy. So it's not very easy to discuss with pharma today. So, but we know we could, again, step by step. We have a partnership with a Spanish pharma. They just lost their reimbursement of their chemotherapy. So we try to work with them to better understand why they lose the reimbursement and which kind of patient do they need to select to be more efficient. So first is to try to better understand who are the good responder. That was my purpose when I say we would like to work with CRO to build ancillary arm to better have the best patient at the beginning.
There's a question at the front.
If I understood well, you first perform a stratification of different patients. How do you plan to evolve this stratification during time? Because you might have new patients. How this stratification will evolve? Or
Can you repeat? Sorry.
if I understand well, you have 3 levels of stratification.
Yes.
So this has been done based on a certain number of patients, and maybe this will evolve with including new patients. Do you plan to do this, or how do you?
Yeah, thank you very much. Sorry. So this is a good point too. What we did first in leiomyosarcoma, we work on a cohort of 112 patients, which is very low number of patients, but we have to take in consideration that is the biggest cohort of patients published. And on this cohort, we took a few patients just to define the seuil. threshold. And then the rest of the cohort was to evaluate the concentration and put in low, medium, or high. Now we're writing for our second publication. It's almost done, and we will be able to, based on the RNA-seq, to just have the result of RNA-seq to say if it is low, medium, or high. So again, that will be less expensive for us, and that will go faster for patients.
Right on time. Thanks very much. A massive round of applause.
Merci.
All right, so thanks very much. For the last startup, we've got Behzad Shemadi, who's the co-founder and CEO of Bright Console. He's dialing in from Vienna, Austria. Can you hear us, Behzad? All good?
Hi, everyone. Can you hear me?
Yes.
Okay.
Okay. Over to you. The stage is yours.
Great. Hi, everyone. My name is Behzad. I'm the co-founder and CEO of Bright Console, and we are on a mission to enhance the global access to safe and effective radiotherapy. Did you know that more than 10 million cancer patients do not have access to radiotherapy? And those who have access go through 15 expensive therapy sessions, which last for 30 minutes each. And sadly, 90% of these patients experience radiation burn after the treatment. But what if I told you that there's a better way of radiation treatment that could reduce these frustration for the patient and also cost for the healthcare system? The answer is flash radiation therapy, which has been tested on the first patient with excellent result. With this approach, patients receive all the radiation doses in one session, which lasts less than a second. Flash can increase the throughput of... Can you hear me?
Yeah, all good. Keep going.
Okay. Flash can increase the throughput of radiotherapy and make it accessible to more patients. It can also reduce the cost of treatment by more than 15-fold and significantly reduce the side effects for the patients, such as radiation burn. Flash is still at the earliest stage, and to be clinically approved, a reliable dosimetry system must be developed. And that's exactly what we are doing at Bright Console by introducing Bright Eye, the first reliable dosimetry system for Flash. Let me show you how it works. A radiation-sensitive patch covers the skin of the patient and lights up when it receives radiation from the source. Then a camera captures the intensity of this light, which actually corresponds to the delivered dose to the patient. And finally, a software can compare the planned dose and the actual delivered dose to the patient and even interrupt the radiation when things go wrong. We have 10 patents around the scintillator or the patch technology, and we are filing a new patent that covers the whole dosimetry system. And we have successfully conducted a freedom to operate analysis. Compared to other solutions on the market for dosimetry for flash radiation therapy, we are the only company that can provide all the features required and requested by our customers. Our business model is B2B, and we are targeting researchers in the first phase and sell our dosimetry system for 70,000 euros, and we already targeted more than 200 research groups around the world. In the second phase, we'll target radiotherapy machine producers and expect to generate 190,000 euros per machine per year. That includes selling scintillator or these patches for 100 euros each, and we expect every machine to require 700 pieces annually. We also charge our customers 120,000 euros for the camera, software, and the service. Currently, there are 18,000 radiation therapy machines around the world. That means there is $3.4 billion market opportunity for us every single year. We team up with the Belgium Nuclear Center to test our technology in the field, and we are part of the preclinical study for flash radiation therapy and focusing on skin cancer. We also received 5 orders from the researchers for our dosimetry system for the future clinical trials, and that could generate 350,000 euros already next year. We already started the certification process and expect to get CE mark and FDA approval by early 2027. And we expect to sell 50 certified dosimetry systems to companies like Elekta and Varian in 2027 and generate 10 million euro in revenue.
1 minute to go.
Less than a fraction of the market. So this is the management team. I co-founded the company with my PhD supervisor, Eric, who is a professor in the field, and we will expand the team with complementary skills when we secure the grant and funds. I also have a great team with me. Spencer is one of the celebrity in the field of radiation detection company and sold his company for $75 million. Hans has 20 years of experience in sales. We have Combis, our first angel investor, and we have Christopher as our scientific advisor from the University of Oxford, and he's one of the pioneers in the field of radiation detection. Since we started, we raised 2 million euros. We are backed by business angels and Austrian public grants, and currently we are raising 6 million euros seed round in 2 tranches. In the first tranche, we need 2 million euros, and of which 1 million euros is already committed, and we will use the fund to get certification, expand the team, IP portfolio, and accelerate the product development. And the second tranche will be 4 million, and if possible, we will mitigate that with grants, and we will use that money to continue the product development, expand the team, and roll up to into the market. We are Brachem-Sol, and we aim to enhance global access to safe and effective radiation therapy. If you have the same vision, join us to help millions of cancer patients. Thank you.
Many thanks, Dr. Behzad. Thanks very much. So let's start with the questions, please. Who wants to go first? The gents on the second row.
Hi, Loïc Vaugier, I'm a radiation oncologist in Nantes. So thanks a lot. I'm impressed by the presentation because it's about flash therapy. First remark is we already have a lot of patients treated with conventional radiotherapy, and it's already quite safe and quite effective. But I agree that flash therapy is very seducing for the reasons you have mentioned. But there are a lot of challenges for this treatment, and we are still very far away from a normal flash treatment for patients in routine. Maybe for skin tumors, as you exposed, and maybe with your detector, but on your business model, you have other than flash therapies on the scope, or flash radiation is the only applications you have, because it's still very for the future, I think, for the future in terms of maybe years. So it's quite challenging, I think, for the development of the model.
Yeah, so you're absolutely right. So it takes some time, and there are a lot of moving parts for flash radiation therapy. But actually, our system could be used for conventional radiation therapy, instead of, for example, films to save times for the team. And actually, we can also get the real-time information about the beam position and also the delivered dose. And there are some solutions like Cherenkov radiation monitoring, and we see our system has some advantages, and not only for flash radiation therapy, but also for conventional radiation therapy.
Okay.
Any more questions?
And I have another one, because you show something very superficial. But what about if you go to flash therapies for example, the prostate cancers or lungs, which are much deeper in the body? What about your system? Is it still working?
Yeah. So actually, so it depends on the penetration depth of the beam. For electron, I think it's around 5 centimeters, but there's some solutions that you can just cut open the body and then expose the cancer tissue. So what we can do, we can measure the skin dose at the moment, but we plan to make a 3D dosimetry system, basically the same composite, but it's 3D, so we can predict what's the penetration dose, what's the penetration of the radiation beam, and then we can also can estimate what's the beam parameters for the deep tissue. But currently we are focusing on the for example skin cancer, where the beam penetration is not the issue.
Thank you.
No more questions? All right, thanks very much, Behzad. Thank you.
Thank you very much.